let us move on to an important topic, alkenones. These are basically used as a biomarker. So we have been seeing about the Arctic freshwater changes and the carbon cycle changes. Now we are actually going to understand how do we know all these things are basically happening. So certain microalgae of the class Pyramnisophisia, notably the marine coccolithophorids, and other living and extinct members of the family Gyphrocapsae, they have had the capability to synthesize alkenones. The main thing is these bacteria could synthesize some chemical compound, alkenones, whose extent of unsaturation, whether 2, 3 or 4 double bonds, change with growth temperature. So this is how, if temperature is high, they produce these particular chemicals. If the temperature is low, they don't produce the chemical. So now we are going to relate how the surface temperature can be measured by simply measuring the chemicals that are being produced by these microalgae and other species. So the unsaturation index of alkenones, this is given a letter U, K37. So 37 is the number of carbons, and U, K prime 37. So this is another one, is defined as, there are some equation, we have to just see what it is. The 37 is the number of carbon that is present. The 37 has 2, 3 or 4 double bond. So that 37 carbon unit has 2 double bond or 3 double bond or 4 double bond. Based on that, we can actually relate that with the surface temperature. So that is what we are going to see. So UK37 is equivalent to the total concentration of C37 having 2 double bonds divided by the concentration of C37 having 2 double bond plus the compounds having C37 having 3 double bonds. So this is the total amount, out of which how much C37 having 2 double bond is present is the value or the index UK37. And when we do that UK prime K37, K prime 37, the K prime is nothing but we added the 4 double bond unit also into the calculation. So here we do the C37 having 2 double bond minus C37 having 4 double bond. So that is the concentration we are calculating. Then divided by the total concentration of all the C37 compounds, whether they have 2 double bond, 3 double bond or 4 double bond. So all put together, that is the total concentration we are measuring. That is U K prime 37 index. So between these two things, we can easily figure it out how these are related to change in the temperature of the Arctic region. So these are positively correlated with the temperature and had been widely used for the paleo sea and lake surface temperature reconstruction. So based on this compound, we can say what was the temperature earlier. So that is what is the relationship. Now here, what are alkenones? The C37 carbon, 37 carbon units we have been talking about, they have the double bond. So what are the different double bond? 2 double bonds. So these are called as C37-2, that is 15E22E, hepta triaconta 1522 diene 2-one. So here is the second carbon, we have the ketone. So that is what this is, having a double bond. So ene, and this is one, and this is an alkyl chain. So alkene one is the name given to this kind of compounds. And similarly, when we have 3 double bond, we have 1 additional double bond here at the 8th carbon starting from this end. And when we talk about 4 double bonds, we also have another double bond at the 29 carbon. So how do we calculate? We start from here, 1, 2, 3, 4, 5, 6, 7, 8. So the 8th carbon, the double bond starts, then 9, 10, 11, 12, 13, 14, 15. So the 15 carbon, the second double bond starts. 16, 17, 18, 19, 20, 21, 22. The 22 carbon, the third double bond starts. 23, 24, 25, 26, 27, 28, 29. So here 29, the fourth double bond starts. And all of them are basically trans double bonds. That is where the letter E is given. E and Z nomenclatures are generally used to represent the geometric isomerism in organic chemistry. So what are the unique properties of this alkenone? These C37 to C39 alkenone are the most refractory lipids known, and they cause the cause of resistance to degradation may be they are low soluble in lipids, or maybe the protective package with algal cells and their unusual trans double bond configuration. So these may be the reason they are not degraded by the other systems. In fact, many bacteria lack enzymatic systems for the degradation or transformation of these kind of double bonds, so they survive for longer time. In general, more degree of unsaturation, the more easily a lipid is degraded. This is the normal thing. But in the case of alkenone, the low number of double bond in relation to the chain length and the wide separation of the double bond, that's what we say 8, 15, 22 and 29 are the distances, like 5, 6 carbons are in between them. So because of the wide separation of these double bonds, the carbon chain suggests that their refractive properties are more or less similar for all the type of compounds. Similarly, the C36X methyl and C38-2 ethyl alkanones have also been proposed as a evaluation of SST, but C37 is the most commonly used chemical for the SST measurements. Why they are unique? There is no isomerization of the double bond. Even if they are stored at 5 degree to 10 degree, there is no isomerization. So that is very unique. And the darkness and adequate wrapping can keep these compounds without breaking for longer time. Similarly, there is no change in the C37 alkenone composition even after acidification, especially for the carbonate removal. Even under ocean acidification conditions, they survive. The microbial degradation of UK'37 is 0.78 or 0.6 under oxic. Oxic means dissolved oxygen conditions. Sulfate reducing and methanogenic conditions showed that these compounds are degraded at a much lower rate than many other lipids. So even the sulfate reducing bacteria or methanogenic bacteria, they don't affect these compounds. Similarly, the UK'37 ratios are not altered as a consequence of diagenetic processes, although the concentration of C37 alkenones decrease. So here we are talking about the 4 double bond species with respect to the rest of them in the faster oxic environment than in anoxic environment. That means they are not easily degraded. So that is the reason they are used as markers for the sea surface temperature measurements. And fatty acids, sterols and alkanols are degraded much faster than these alkenones. So that is the reason, and these alkenones are known only in the Arctic region. So that's the reason they are used as a marker to find out the temperature difference. And the percentage of C37,4 alkenone relative to the total C37 alkenone is in the northern high altitude waters and surface sediments are found to correlate strongly with annual mean sea ice average. So that means there is a relationship. So from the mean sea ice coverage, we can actually see that it is also related to temperature. If the temperature is high, obviously ice will not be there. So that means this 4 double bond species may be related to temperature or may be related to mean sea ice also. And similarly, the salinity level had no impact, literally no impact on the C37,4 type of compounds. So these alkenones are not at all affected by the change in salinity level. So that means there is no physiological effect on salinity. So they survive for longer time without any degradation. So, but we still do not have a consistent evidence to selectively biogenically degrade the C37,2 and C33 alkenone by biota. So, even the other things like 2 double bond system and the 3 double bond system are not selectively degraded, but of course they survive. That is the main thing. And the 4 double bond one is directly correlated to temperature in a much better way. So globally, in most cases, the UK37 ratio, which does not include the 4 double bond species, will be a better proxy for paleo temperature estimate than U4'37. This is in the normal region, every region. But in the high altitude, especially in the Arctic region, this C37,4 alkenone is a very important one for identifying the paleo temperatures. So here is the flowchart for the collection of ocean sediment and analysis for alkenones. So how do we do that? we do the site selection first, then we go to the sediment sampling. We collect all the sediment samples, and especially the sediments we collect. From there we have to extract the alkenone and find it out how much it is present. That is as simple as that, but the whole process is given here. First we do the core processing. So we do two type of analysis. One is a chemical analysis, another one is a physical analysis. In the physical analysis, we determine the sediment composition. So what are all the different type of materials that are there, that we'll see. Size wise, shape wise, those kind of physical parameters we will check. Then we also assess the stratigraphy. So how much they are distributed, that also we will see. In the case of chemical analysis, we extract the lipid from the sediment sample. So we use some solvents and then extract the lipids from that sediments. We also analyze the alkenone compounds using GCMS so that we can be accurately knowing how much or which type of alkenones are present, that we can do. Then we quantify the alkenone concentration, then determine the temperature and productivity estimates. Based on that, we compare with the historical climate data, because this is not the only thing we are doing. There are also other studies people had carried out to know how much or what is the temperature in the past. So we can use this alkenone to correlate and compare with the known result values from the other experiments, and we can clearly say, yes, this is what is happening. No, this is not, there is some change we have to do. So those kind of comparison we can do by the alkenone analysis.